Chapter 6, Metabolism and Enzymes. So the main student learning outcomes in this chapter is for us to understand what metabolism is in the human body. We'll try and understand different types of chemical reactions where enzymes are involved. It can either be anabolic or catabolic, endogonic or exogonic. We'll also talk about how enzymes work, some of the features of enzymes, the factors that affect enzyme activity. So our next class is to study cellular respiration. So we will touch upon the structure and function of the energy molecule in our body, adenosine triphosphate, ATPs. Here's a nice animation of an enzyme doing its thing in the human body. So this is a very interesting picture of all the chemical reactions that has been discovered in the human body. So this is a chart in the biochemistry department. So metabolism is all the chemical reactions that's happening in the human body in every cell. I know it's super complicated. So how is it possible to accomplish such a huge feat? It's because of this unique molecule called enzymes. So if you see the reactions that can happen in the human body, it can either be anabolic or catabolic. So anabolic means they require energy to synthesize a large molecule. So you can see on the picture on top where monomers are strung together using energy to form a large molecule. So it's building up. Catabolic is the opposite. Catabolic is breaking down of a polymer into its monomers and releasing energy. So enzymes are going to catalyze either an anabolic or a catabolic reaction. And both of them are required to maintain homeostasis in a cell. And the picture here is showing about all the organisms that can make their own food are called autotrophs. And organisms that, like us, who can't make our own food, depend on autotrophs for their food. They are called heterotrophs. So if you take the environment, there's producers and consumers. Plants are the producers and we are the consumers. So the chemistry, the metabolism in both the autotrophs and heterotrophs are similar and still different also. How does energy flow in the ecosystem? So the main source of energy for all life on Earth is the sun. So energy enters the ecosystem as solar energy. So the producers are going to convert the solar energy into chemical energy. They are called producers. And the consumer will eat the producer. And the byproduct would be heat that's given off to the environment. And then when the consumer dies, it will be decomposed and that will give off heat also. So this is the energy cycle in the ecosystem outside. So this is the law of thermodynamics. Energy is neither created nor destroyed. It's just converting from one form to the other. Solar energy becoming chemical energy in plants. And we take that chemical energy to make ATP energy. And then we decompose and then that goes back to the environment as heat. So we see that cycling of energy in the environment. So energy transformation and thermodynamics is the driver for enzymatic reactions. So the first law of thermodynamics says that the total energy in the environment is constant. It is neither created nor destroyed. It can only be transferred from one system to the other. The second law states that the total entropy of a system will never decrease. Enthalpy refers to the heat that is pertaining to a system. Entropy is the randomness it creates in a system. Gibbs free energy in a system is the amount of measurable or usable energy in a system. So for heterotrophs like us to survive, in order to carry out our bodily functions, our energy comes from plants that provide us with glucose. So these pictures are showing the energy the energy of motion. Kinetic energy is the energy associated with a body in motion. What is an exergonic reaction and the endergonic reaction? So Gibbs free energy is the energy that is associated with the chemical reaction that is used for doing work. So here is an example of an exergonic reaction. And here are the products. So the reactants. Before it becomes the product in an exergonic reaction, energy is released. So this is the rate of the reaction over a period of time. So before the reactant becomes a product, energy is released. So that is an exergonic reaction. So here, Gibbs free energy is less than 0, and this reaction is spontaneous. The endergonic reaction, on the other hand, requires energy for the reactant to become a product. So here the reaction is not spontaneous because the change in free energy is more than 0. So again, in this graph, the rate of the reaction is plotted. So for the reactant to become the product, there is an energy barrier that it has to undergo. That is called as an activation energy. For any reaction to happen, it needs activation energy. So how enzymes work is by reducing that activation energy. So you can see it here. So what is the nature of enzymes? Enzymes are defined as a biological catalyst. Catalyst means something that will speed up the reaction, but it is not consumed in the reaction and it is reused. It is usually a protein, like a globular protein. Enzymes work by lowering the activation energy of the reaction. How are enzymes named? See, like in a word, there's prefix and suffix. So if it ends with ASE, it is an enzyme. And it is named after the substrate it works on. So the reactants are called as substrates in an enzyme catalyzed reaction. So here's an example of sucrase is the enzyme because it ends with ASE. That is going to break down on sucrose, which is the substrate. Proteases will act on proteins, lipases will act on lipids, lactase will act on lactose. So remember, protein is a biological catalyst. It's speeding up a chemical reaction by lowering the activation energy, and it is not consumed in the reaction. It is reused. So that's the picture here that is showing. This is the energy barrier for the reactants to become products. And when an enzyme is present, it reduces the activation energy. So let's look at some of the features that are found in an enzyme. So it is a protein, usually a globular protein. So in the protein, so this is the protein here. There is a sweet spot for the substrate to come and anchor. This is where the reaction happens. This is where the catalysis happens. So that is called as a what? Can you guess? Active site. So in the enzyme, the substrate will come and anchor, and in the active site is where the reaction happens. So the active site is going to provide the right environment for the reactants to become products. So this is where the bonds are either made or broken. So here's an example of an enzyme in gray. The substrate is coming and anchoring in the active site. It is called an enzyme substrate complex now. And this is where the bonds are either being made or broken. This is providing the right environment for the chemical reaction to happen, and the products are released. Here, the last picture here. And the picture on top is an interesting analogy. Like, it's very hard to cycle up a mountain, but if that mountain was like fairly like a bump, it's very easy to go over the mountain. So this fit between the substrate and the enzyme is called as the induced fit model. It's the lock and key hypothesis. See, can your house key open my house? No, right? Because just like how a key is specific to a lock, an enzyme is specific to a substrate. So that is called enzyme specificity. So when the enzyme and the substrate form the union to form the enzyme substrate complex, they will mold around each other. It is called as induced fit. The induced fit model explains how enzymes and substrates will undergo dynamic modifications during the transition state to increase the affinity of the substrate to the active site. So how are enzymes regulated? So there are so many enzymes in the human body, so many chemical reactions. So our body has a way to regulate it. It's by inhibition. So competitive inhibition, a molecule other than the substrate will bind to the active site. Because the active site is already having the inhibitor, the substrate is not able to come anchor in it. And because of that, the reaction can't happen. So that is called competitive inhibition. If the molecule anchors in a different site in the enzyme, thereby changing the active site, it is called non-competitive inhibition. So it's not competing for the active site. So allosteric inhibition is a type of competitive inhibition. Here, the activator molecule will modify the shape of the active site. So that is called allosteric. So here is the inhibitor. By anchoring to the enzyme, it is changing the active site. And because of that, the substrate is unable to come and anchor. So that this picture is explaining enzyme regulation. It can either be competitive or non-competitive. Competitive is when the foreign molecule sits on the active site, and thereby the substrate can't sit and the reaction can't happen. Non-competitive is when it sits on a different site and changes the shape so that the substrate can't anchor. So what are some of the factors that affect enzyme reaction? Temperature, pH, enzyme concentration, temperature, substrate concentration, and so forth. For every enzyme, there is an optimal temperature where it will have its most effect. So for example, most of the enzyme, all the enzymes in the human body will act best and function best at 37 degrees centigrade. And also pH. You know, there are certain enzymes that's in saliva, which is functional in pH 7. The enzymes in your acidic stomach, the pH is like around 2. Whereas if you go to the small intestine in digestion, where absorption happens, the pH is alkaline. So different enzymes have different pHs. So the effect of temperature and pH you have to understand, because extreme temperatures can change the active site of the protein. Another important, interesting tidbit here is many vitamins are required for an enzyme to function. So they're called coenzymes. So a coenzyme is an organic molecule that is going to bind to the active site of the enzyme in order to help the enzyme to anchor. So like here's examples of vitamin A and some B's and D's that are coenzymes. They are speeding up the chemical reactions and helping the chemical reaction. So next week, we are going to study a very important metabolic pathway. So we have to understand what ATP is. It has a 2-ring of adenine, a 5-carbon sugar, and 3 phosphates. ATP is required for energy coupling in a cell. And of course, energy is required for, see here, for the flagella to move or for a sodium-potassium pump to work. So what happens is by the hydrolysis of one phosphate group, ATP can become ADP. And in order to create ADP, you have to like phosphorylate ADP. And the energy will come from food. This is called cellular respiration process that we are going to study next week. So this is the structure of adenine triphosphate. And ATP is the energy molecule that couples reactions. Usually the endogonic reaction and exogonic reactions are coupled so that they can happen in the cell. So here's a metabolic pathway. So it is a series of reactions. So for example, for product A to become like product B, it needs enzyme 1, and then enzyme 2 for the next step, enzyme 3 for the next step. And how is the whole pathway regulated? Many a time when there is a build up of the end product, it will act as an inhibitor of the enzyme earlier in the pathway. So that is called feedback regulation.